Welcome to Mechanisms of Cancer. Today we're going to be talking about RB and E2F. So let's get a little bit of context first. We're talking about a cell moving through the cell cycle, a cell that would be dividing, and it's going to be moving from G1 or GAP1 phase into synthesis. In order for synthesis or DNA replication to occur, a lot of proteins have to be expressed, and a lot of those proteins are expressed in G1. We're talking about a specific set of those proteins, specifically MCM, which is helicase, ORC, which is going to be the origin of replication complex, and a few other synthesis-related genes, all of which are activated by a single transcription factor. And that transcription factor's name is E2F. So the basic story is simple. E2F, a protein, binds to a promoter on a sequence of DNA, recruits RNA polymerase, and the gene immediately following that promoter is transcribed. In this case, those genes are, again, ORC, MCM, and some other synthesis-related proteins. Now, E2F has an inhibitor, a natural inhibitor named RB. It was named after a disease in which it was found, and we'll go into that a little bit more. But basically, once RB is bound to E2F, E2F is not free to bind to DNA. So if RB is bound to E2F, then the synthesis genes are not expressed. That might seem like a bad thing, but if you think about it, it actually works out well. Most of the cells in your body are not dividing. So you'd only want to trigger division or synthesis of division genes in cells that are dividing. And RB is one of the mechanisms by which the body or the cells in the body accomplishes that. Now, RB inhibition of E2F is reversible. That means that you can turn off RB. Simply by adding a bunch of phosphate groups to RB, you can change its shape, and that shape change allows it no longer to bind to E2F, which means E2F is free, again, to bind the promoter and cause expression of those genes. Now, that phosphorylation is reversible. Your body can phosphorylate RB and dephosphorylate it. RB was found, however, because of the disease retinoblastoma. And in that disease, RB is actually mutated. Now, RB is mutated in such a way that it can no longer bind to E2F. Now, you have two copies of a gene. You might have either two mutant RBs, a mutant RB, and a regular RB, or two regular RBs. But if you have two mutant RBs, that means that E2F is always binding to the promoter of those genes. That means ORC, MCM, and other synthesis genes are always being made, and that means that certain cells in your body are proceeding from G1 into S without being told to. Now, that's not such a good thing. So what's important about this story? Well, there's a couple important things. The idea of pathway is really, really important. So this is an example of one thing triggering another thing, triggering another thing, triggering another thing. RB inhibits E2F. E2F activates expression of certain genes. Phosphorylation of RB inhibits RB, which will then activate E2F. So let's think about that really quick. It's like RB is an inhibitor or an off switch for E2F. And what you're doing is when phosphorylating it is turning off the off switch. So it's kind of a double negative, but you're going to see examples of this in the body over and over and over and over again. These kinds of mechanisms allow for several layers of control. So you can control E2F, you can control RB, or you can control the thing that's inhibiting RB by phosphorylating. At the end of the day, it's one of the first examples of a checkpoint in the cell, a way that a cell stays in a certain state before moving forward, and that way that cell stays in that certain state is by binding E2F to RB. That's it for RB and E2F. It's a pretty simple story, and good luck on understanding it.